Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Rabbi zidni ilma, Rabbi zidni ilma, Rabbi zidni ilma walikni bis-salihin. Allahumma inni as'aluka ilman nafi'an wa rizqan tayyiban wa 'amalan mutaqabbalan. Assalamu alaikum students. Today you are going to learn about the third part of the lecture, which is the structure of the proteins, what are the properties of the proteins, and what is the denaturing and coagulation differences of the protein, and what is the importance of the protein. This is your biochemistry chapter 21 part 3. Now the discussion is about the structure of the proteins. The building blocks of the proteins are amino acids. The basic one is amino acids, which are small organic molecules that consist of an alpha central carbon atom linked to an amino group, a carboxyl group, and a hydrogen atom, and a variable component called a side chain. Two amino acids. On the right side, if you observe over here, this is the basic alpha carbon. Alpha carbon is going to be connected with on the left side, it is going to be connected with the amino group, NH2 group. On the right side, if you observe, this is COOH, carboxylic acid or carboxylic part, and in the center, it is connected with the alkyl group and along with the hydrogen. So basically, this is called the amino acid. This is the monomer of because it is going to be converted into the polypeptide chains, and the peptide linkage is that when two amino acids molecules are attached to each other through bond, that bond is known as the peptide bond. If you observe over here, this is the peptide bond, this is the amino acid 1, and this is the amino acid 2. When H2O hydrogen, the water is going to be loose over here, and this is going to be connected with this part is going to be connected with this part. You know that in this way they are going to be connected with each other. This is called the peptide bond. In this way they form the long continuous chains. The polymerization will occur. So its monomer is the amino acids. So this was the start of that, and basically there are three types of the combinations of the structure of the proteins. One is called the primary proteins, then the alpha helix, the beta sheets are there, then the tertiary one, and the quaternary one. So one by one we are going to observe these structures. First one is the primary structure. The sequence of amino acids in a polypeptide or protein molecule is primary structure. NH3 group is going to be an amino end is there. All, if you observe, this is just like the beads are connected with each other, just like the necklace. This is the amino acid subunits. What type of the combinations are there? These all on the right side, amino acid, then their abbreviations are also given. These are the alanine, arginine, asparagine, aspartic acid, and the cysteine, the glutamic acid, the glutathione, the glycine, histidine, isoleucine, leucine, and lysine, methionine, phenylalanine, proline, serine, threonine, tyrosine, tryptophan, and valine. These all are the amino acids, and in a special type of the symmetry, they are connected with each other, just like the beads. In this sequence, when they are arranged, this is called the primary structure of the protein. Now, the secondary structure, the way in which the primary structure of the polypeptide chains are folds. Here, observe, this is the foldings when they are going to be folded with each other. The subunit is amino acids. The beta, there are the two types of, one is called the alpha helix structure, the other one is the beta pleated sheets. These are the beta pleated sheets. After synthesis, polypeptide chains are folded or pleated into different shapes: alpha helix, regular 3D shapes, and the beta pleated sheets, the twisted pleated sheets. Here, right side, twisted pleated sheets. The helix is held by many hydrogen bonds between amino acids at different places in the chain, giving the giving the chain the great stability. So, in this way, when they are going to be folded and pleated, this type of the shape is called the secondary structure of the proteins. Next one is about the tertiary structure. The long peptide chain of the protein molecule undergoes the folding and refolding on itself and gives rise to the structure known as the tertiary structure. So They are just like this. If you observe, just it look like this. And when into the chemistry of when we are going to observe, there is the folding and the refolding is hydrophobic part interaction. The polypeptide backbone is there. If you observe over here, the hydrogen bonding is also present in between the molecules, and disulfide bridges are also there, and with the ionic bonding. So in this way they will be refolded and folded. So when the protein is going to be refolded and folded on itself, then this structure is called the tertiary proteins. Then the quaternary proteins, the quaternary structure is look like this: the aggregation of the two or more peptides. When two or more peptide chains, for example secondary and tertiaries, when are arranged into a large size molecule from the quaternary structure. So this is called the quaternary structure of the proteins. Now we are going to discuss about the properties of the proteins. In the properties of the proteins, the color and the taste. The proteins are colorless, usually tasteless. When the shape and the size of the proteins range in shape from simple crystallite spherical structure to long fibrillar structures. Molecular weight. They are complex organic compounds with high molecular weight. They are basically amphoteric in nature because they contain both the acidic part, COH, the carboxylic part, and the basic NH2 amine group. They react with acids and bases. Then we are going to learn that your property next coming is the difference between the denaturing and the coagulation. Denaturation is a process in which the proteins or nucleic acids lose the quaternary structure, tertiary structure, and secondary structures which is present in their native states. So when they lose their structure on that time, this is called the denaturing. And coagulation is defined as a change in the structure of protein from a liquid form to the solid or thicker liquid brought about by heat, mechanical action, or acids. Enzymes may also cause the protein coagulation. For example, is that the cheese is going to be the example of the preparation of the cheese is going to be example of the coagulation. So the importance of the protein is that they are the building material. They maintain and repair all the body. Your head to toe, you are made up of the proteins. Your hairs, your nails, your skin. So they will build and they will maintain and they will repair the all body cells. Number two, they supports the healthy bones and muscles and skin growth. Number four, then they supports the healthy eyes and vision. Supports a strong immune system is also related with the importance of the protein. So here are the diets which we can use, which are rich the fruits and the vegetables and the all these are also contains, these are called the protein rich fruits. So nature can help you with the maintenance of the food and giving this so healthy body to you. So take care and eat the healthy proteins. It will not increase your weight also. That today's conclusion is that today you learn about the structure and properties and importance of the protein, and in next lecture inshallah we will learn about enzymes. May Allah be with you. Take care. Allah Hafiz.